Fine. Should I start them?
Give a couple of seconds.
Okay.
Yeah, go ahead.
Okay. Good morning, good afternoon, and good evening to all of you who've joined us today on this edition of Biologically Speaking and its webinar series. So today's theme is the synapse. And we're joined by two eminent neuroscientists. The first, Professor Upinder Bhalla from NCBS Bangalore, and the second, Dr. Poonam Thakur from IISER-TVM. They both study the brain and how synaptic connections between different types of neurons and different areas of the brain lead to behavior, memory, and on the other side of the coin, also to neuropathologies. So for the audience to know, please post your questions in the chat box when the talk begins. We will take a few questions after each talk, and but in the interest of time, we might have to move on to the next speaker, but we will definitely take your questions at the end when both speakers have finished their presentations. So without further ado, let's move on to the first speaker of this session, the synapse. The first speaker for today is Professor Upinder Bhalla. We're delighted to have you here, and it is my utmost pleasure to welcome Opi to this platform, not only because he's an eminent neuroscientist, but also personally because he happens to be a mentor I deeply admire and look up to. So Professor Bhalla graduated in the natural sciences from Cambridge University, following which he pursued his doctoral studies in the lab of James Power at Caltech. He graduated in '93 and moved to Mount Sinai College of Medicine, School of Medicine, where he did his postdoctoral research in the lab of Ravi Iyengar. On his return to India in 2002, he joined the National Center for Biological Sciences in Bangalore, where he has been since. Upi's lab has been a pioneer in the field of olfactory coding and studies mechanisms of memory formation in the brain, and his research spans neuroscientific techniques ranging from 2-photon calcium imaging to patch clamp electrophysiology, as well as computational modeling to answer these questions. Professor Bhalla, as you all know, is also the recipient of numerous scientific awards, among them the Shanti Swarup Bhatnagar Award in 2007, and more recently, the Infosys Prize for Excellence in Research in 2017. We are again delighted to have you on board at Biologically Speaking, and over to you. Hope this is an amazing talk.
Thank you so much, Shreya and other colleagues from Biologically Speaking. And like Shreya, I'm very glad to have everybody here in the audience from whichever time zone you may happen to be. I'll just share my slides and then we will go from there. So I trust that you can see the slides.
Yes.
Okay, good. So it's a special pleasure to be here because, as Shreya says, I had the good fortune to have her in my lab for a short period. In other words, it would have been nice if she would have been willing to stick around and go on to a degree. But anyway, it was a great pleasure to have her in the lab, and I'm therefore very happy to be at a presentation which she is helped to organize. So what I'll do is I'll give you a very brief overview of the topic that really fascinates me, the study of memory. How do we remember things, and where do we go to look for it? And I'll give you a sense of a couple of projects that we have undertaken over the years to try to get at least some glimpses of an answer to this. So let me start with this very standard, old-fashioned, and yet absolutely pertinent point, which is that the brain functions at many levels of computation. And though what we see when we interact with each other, with animals, with the world, is at the level of behavior, the computations that underlie it actually span all of these levels, all the way from molecules, synapses, dendrites, neurons, circuits, areas and systems to give rise to the behavior. And we know perfectly well that molecular changes such as mutations can give rise to behavioral outcomes, but we're also learning that the converse is true. So I've always felt, and I'll try to give you a glimpse of this in this talk, I've always felt that it is essential to, when studying the brain, to be very aware of the constant flow of information across these levels and not to fall into the trap of focusing entirely on one level or the other. Because if you do so, you will miss some of the, to my view, some of the absolutely crucial aspects of how the brain functions. So with that intro, let's dive right in. So what I'm going to do in the next few slides is to just walk you through the various scales of examination of brain function. So this is, of course, our favorite organ, or as Woody Allen said, it's our second favorite organ. Knowing Woody Allen, I'll let you figure out what he meant. But the point is that this organ is about 1.4 or so liters in volume. It takes about as much power as a reasonably efficient light bulb, and yet it does everything that makes us uniquely human. So how does it work? So this is the level at which, of course, we generate the final outcome, the behavior. So now what we're going to do is to zoom in from this centimeter, tens of centimeter scale object to that little blob over there in the corner, which is the size of a neuron. And here we have a computer model of a neuron, roughly 500 microns from end to end. This is a Purkinje neuron. It's actually a model of a Purkinje neuron drawn from some work by my friend and colleague, Eric de Schutter. And so this is a very important scale at which a lot of analysis is done. And most recordings that you will see typically come by sticking an electrode in the cell body over here of this cell or of other cells and figuring out what activity it undertakes. But we are going to now keep zooming in. And now we're going to zoom into the small volume around here, which is the volume of a small piece of dendrite. And so we're going down to 5 microns now, 100 times smaller. And so this little cube of tissue was serially EM reconstructed by Kristen Harris and co-workers. And this yellow object in the middle, branched object, is one piece of dendrite, a small piece of dendrite. And these little knobs and protrusions coming out of it are postsynaptic densities and postsynaptic spines. And the little red marks you see are the locations of the synapses, of the junctions between neurons. And the size of a synapse is given by this little dot here. And as you can see, there's a lot of them. In fact, the neuropil is incredibly packed. It's completely jam-packed of connections and nervous tissue, dendrites, axons, and so on. So let's zoom in one more time to the size of a synapse. And this beautiful drawing, or I should say, it's a kind of a computer model, takes us down to the size where we start to see molecules doing their thing. So this is half a micron across, 500 nanometers. It's the presynaptic bouton, and there's the release site down there. These little bump-like things you can see on the surface, those are surface proteins. And the little strands and globules and all you see in here are individual protein molecules. The little round things are, of course, vesicles. Here are some microtubules. So we're now really talking about molecular scales. And the point I want to make is that computation is happening, memory is happening at all of these scales. And we'll get into this in a little bit more detail. So let's back up, though. Let's go to the classic studies of Ivan Pavlov, which you're all familiar with, about how we form memories, right? And this is such a classic thing. In fact, some of the experiments I'll tell you about use pretty much the same kind of idea. So just to recap, something you're all familiar with, what he found was he was actually interested in digestion and salivation, but he was a sharp scientist. He noticed that when the dogs were presented with food, not surprisingly, they salivated. But because his was a well-organized lab, there was a bell that rang every time food time was to come around. And after some repetitions of this, the dogs started salivating as soon as the bell came on. And in due course, they salivated just to the sound of the bell. So they had learned something. They had formed a memory or an association between the sound of the bell, which is a neutral stimulus, and the food, which is very much a not neutral stimulus. And so this is one of the basic paradigms for learning and memory, one of the basic approaches. And as you'll see, this is something that we also have used. So let's now imagine that we have a dog with 3 neurons. Yeah, this is a cartoon dog. And what I'll try to do here is to draw the link between the behavior, which is sort of the very top level emergent phenomenon of how the brain is doing all this stuff, all the way down to the molecules in this cartoon 3 neuron dog. So this this dog, it observes food and it's got a food neuron. Haha. So there's the food neuron, and what the food neuron does is it causes the dog to salivate, right? So it sees food and it salivates. So that's nice and straightforward. But there are 2 other neurons that this dog has, one of which receives sensory input from, say, a bell sound, and another one which may receive, let's say, light. And there's only one difference between these 2 stimuli, right? Just like in Pavlov's dogs, the lights were on. Yeah, there was day-night cycle. But the only difference is that there is an association between the bell and the food, but there's no particular association between the light and the food. So what we imagine happening is that when the bell goes off at the same time as the food neuron is active, there is sort of repeated association, and what was a weak synapse, a weak connection between the 2, over repetitions, over many cycles, becomes larger, becomes stronger, so that in due course, when you trigger the bell stimulus, this connection is strong enough to trigger the salivation. And so the learning has happened here at the connection between the 2. And how that learning happens, how the connection changes, is the basis of the molecular part of our study. In contrast, the control is that the light had no particular association. And so whatever synaptic input it had, whichever weak input it had, does not change. It has not learned that the light has anything to do with it. So this, in a nutshell, is a 3 neuron version of that takes you all the way from the behavior, which is the ability to learn that the bell implies food, implies salivation, all the way through the cellular physiology, the synaptic physiology, and the molecules that underlie it. And this is a cartoon version of how all of these levels may be connected. Okay, so let's move on with this as a kind of a cartoon version of learning processes. And of course, there are much more elaborate understandings that we now have. I won't go into those. So here's another way that we have looked at memory. And this is the tragic case of Henry Molaison, a patient HM, who had the misfortune of having intractable epilepsy, following which the surgeons removed this large chunk of tissue, which happened to include the hippocampus. Yeah. So Brenda Milner, among others, looked at this patient for years, literally for decades. And what happened was this patient lost short-term memory. He could not form new memories, yet he retained long-term memories. In other words, he remembered what had happened to him before the surgery. And remarkably, he was still able to form new procedural memories. So for example, he was able to learn new skills. Procedural memory might be something like learning to ride a bike. So he could still do that. Not to say that they taught him to ride a bike, but they taught him things about drawing and so on. Even though he had no recollection even of having met the doctor the previous day. So there are different kinds of memory in different locations in which they're stored. And clearly the hippocampus has something to do with the short-term memory. And that is where a lot of attention in the field goes. And that's what I'll be talking about. Okay, so what's the hippocampus? The hippocampus in your brain is this somewhat banana-shaped, or actually seahorse shaped, that's where it gets its name, structure in red over there. And it does many things. For example, in many animals, including us, it is involved, among other things, in remembering places and their association with events. So this is work for which the Nobel Prize was given not too long ago for finding the establishment of place cells to bliss and of course my memory fails at this point, but in studying the effect of place on activity of these cells. So the animal remembers where it is, and its manifestation of that is by the activity of cells which learn to be active in a certain location. So what we've done is we've sort of gone down various levels. We've gone from the systems level, hippocampus and amygdala, which is involved in other kinds of memory with emotions. We've gone to the area level, which is subdivisions of this. Cell and circuit level involves connectivity changes and excitability changes. Synapse level involves conductance of receptors. Now we're talking at this scale. And at the molecular level, we have things that we'll be looking at, switches and other changes. So where my lab looks at is the hippocampus. We look at various kinds of circuit effects and area effects, and we look at synapse effects and molecular effects. And I, of course, will only talk about a couple of these. Okay, so that was a long introduction, but I felt that it's interesting to get to see what is the domain of what we work on. So I'll talk now a little bit, just a glimpse of what we do at the hippocampus level, looking at recurrent and associative networks. So what we do here is we work in the model system of the mouse. And the way we do this is this relatively recent, as in about a decade or so, technique of optical imaging using genetically encoded reporters. We make small windows in the skull, so that's the location of the window. This is a top-down view of the skull. And you can actually see the glowing green color of the fluorophore, of the reporter whose fluorescence changes when the cell is active. Here's a cross-section of the brain, so you can, of the same brain. So you can actually see the region which was illuminated and which is, in this case, expressing a dye. In more recent versions, as I said, it's genetically encoded. So this is the hippocampus. And we do these recordings using custom 2-photon microscope. This is the new one, who my students have called this new machine Black Widow. The previous one was called Bheja Fry, which I think is a marvelous name too. And this is an image that we get from this microscope. But let me show you what the image actually looks like after I explain how it does the learning. The way it does the learning is we're doing a trace conditioning task, which is very, very similar to Pavlov's task, which is that there is a conditioned stimulus, in this case, either a sound or a light. And there's an unconditioned stimulus, which can be an air puff. That's the air puff to the eye. So the animal blinks when an air puff comes to its eye. Or it can be a stimulus to the paw, a small zap, which causes the paw to tingle, and then the animal removes it. So we have this association, and they're separated in time. So here's the sound or light, and here's the air puff. And what happens is that after a few repetitions of this, just like Pavlov's dogs, the mouse learns that, oh, I heard something, therefore I should close my eyes, or I saw a flash of light, therefore I should close my eyes before the air puff comes. And that's what the animal learns. And this is the training protocol that we use. This is a very multimedia kind of experience for the mouse. So there's the mouse. You can just about see its backside with its tail protruding on a little treadmill, which we've got. The experiments are done in the dark because we're doing 2-photon imaging. We can't have stray light coming in, so we have infrared illumination so that we can watch what the animal is doing. We have different kinds of stimuli for the light, for the paw, for the sound. We have the air puff. And so that's generally what the experiment looks like. Of course, this is all placed inside a 2-photon microscope, which is focused on the animals, the window in the animal's head. And this is what we see. So these are some of my earliest, or rather my lab's earliest images. And you can sort of see cells flashing on and off. This is while the animal is actually learning something. We had a power outage, but I think you can still hear me. So anyway, so here we have the flashing of the cells which represent brain activity, which is what is happening when the animal learns something. So this is tremendously exciting, but what it means for us as scientists is that we can record what the brain is doing while it is learning something. And so now what do we do with this? So here then is a nutshell of one of the key research programs we're looking at, which is to look at the life cycle of a memory trace in the hippocampus. And there are several questions, and I'll only briefly give you a glimpse of a couple of these. One of them has to do with formational memories. How do individual cells take up representation of the stimulus, and how do they represent the interval? There's competition between different stimuli. There's coexistence between different memories. How do you manage to remember so many things at the same time? And then, of course, there's the process of forgetting. And we're looking at all of these in our experiments. I'll mention a couple of aspects of this. The others are very much active research. So here's a study that we did some years ago now, Mehrab Modi and Ashish Bhalla. And they looked at the formation process. So what you have here are just 6 cells out of about 100 that Mehrab recorded from. And each cell, we're showing 3 trials. And we're showing trials before learning and after learning. What you have here is the fluorescence level. That's what the 2-photon microscope is measuring. And the little asterisks indicate the time to peak. That means the time when the cell activity was largest. And I think you can see that, and okay, down here you have the training stimulus, and there you have the air puff. Okay, so this is the activity of the brain represented by these 6 cells over 3 trials during learning. And before learning, you can see activities all over the place. There's no particular correlation. After learning, something really remarkable happens, which is that not only do the cells all start to line up, they form a sequence of activity. And so this addresses the first point I brought up. How do the individual cells take up representation of the stimulus? Well, they do that by converging their activity to a fixed time point. And they represent the interval by sort of tiling, by spanning, by bridging the interval between the stimulus and the unconditioned stimulus. So this is one aspect of our study of the life cycle of a memory trace. I'm going to skip over a couple of the others, but here's some very recent stuff that we've been looking at, which is how do you have coexistence of 2 forms of information? And so what we've got here, so here's again the same view of the hippocampus. Those are the individual cells. Here, what my student Shomo has done is to color code the cells by their responses. And red means that the cells were not selective for any stimulus. Green means that they were selective for sound. Dark blue means that they were selective only for light. And the cyan is the overlay, that is, they were selective both for light and sound. So this is how you can represent 2 different stimuli which the animal has learned at the same time, in the same mouse, in the same set of cells. So some represent nothing, some represent one stimulus or the other, and some represent both. So that is how you can have coexistence of memories. And there's lots of theory that goes into that. Okay, so this is the glimpse of what we're doing on the whole animal level. And let me just now shift gears. I won't mention much about the circuit level stuff, other than to say that we have our hippocampal slice here, and we stimulate it optogenetically using the wonderful tools that of Chandler-Dobson that have come out of Carl Desserot's and other groups. And we use this then to probe the memory and other computation in the brain slice. But let me now dive very briefly into the molecular level, where we look at switches, structural change, and relocation. I'll just give you a glimpse again. So just to remind you, these are all the molecules that do this lovely computation. This is what it looks like in the computer model. That is one of our major tools. This is a very, very old slide, but it just gives you an idea of how complicated even simple models are of these systems. Here's a quick glimpse of a simulation where we're looking at activity on these dendritic spines and what it does to the signaling underneath the surface of the cell. So here you can see the activity is building up with a stimulus of a certain kind. And here the activity on this side fails to build up because the stimulus is not aligned in time. So these are just to indicate the kinds of simulations that we do. Here to remind you then is that in this scale of our 3 neuron Pavlovian conditioning, we're looking at the molecules inside the synapse. And what I'd just like to leave you with is the idea of molecular switches that can flip between one state and another state. You can think of it as kind of 2 energy wells. And the way that you can imagine this happening is that supposing you have a feedback loop of molecule A, which stimulates B, and conversely, B stimulates back A, and you have a pool of such molecules. If you give it a strong stimulus, you can turn it into a state where both are in a high active state. They're stimulating each other. And so they sustain activity. And the reason why this is important is because if you have such a self-sustaining system, it is resilient. It resists changes, it resists memory loss, even when you have molecular degradation, followed by fresh synthesis, leading to molecules which do not, quote unquote, remember what has happened. The new molecules then become part of the feedback cycle. And so your memory carries on, even though the molecules involved lose their integrity. So this is one aspect of molecular learning and memory, which we are being very interested in. Another one has to do with the fact that at very small sizes in the synapse, you have a lot of molecular noise, chemical noise, arising from thermal perturbations and the fact that reactions are actually bidirectional and are composed of individual molecules bumping into each other. So there's a certain probability of them going forward and some not. As a result of which, your nice clean feedback cycles, which store memory, can become extremely noisy, as are indicated in these two blue traces, contrasted with the sort of classical version, which is the red trace. And so how do you store information in a system that looks as noisy as this? And this is something that we have also analyzed over many years. And the short answer is that there are changes, there are kinds of molecular circuits, especially those which include structural change, that are capable of storing information for years, which is what you want to do in memory. And these are all happening at the chemical signaling level. Okay, so to wrap up then, where do you look for memory? I would say you look at all of these levels. You look at the behavior, the circuit, the cell, and the synapse and the molecules inside it. And I've tried to give you a glimpse of how we do this using behavior, using whole animal level recordings from the hippocampus in an intact animal, using models at molecular scale, and using experiments and simulations at the circuit level. So this is an old slide of the lab, and I just wanted to point out that there's Shreya in there. And I would like to thank all of you for listening. I'd like to thank the team for hosting this meeting and all of my funders. Thank you so much.
Thanks, Upi. That was a wonderful talk, covering all the different aspects that the lab works on, basically. I'm a bit sad that Bheja Fry has moved on to Black Widow somehow. I have a lot of good memories with Bheja Fry. I think we have few questions, and if...
Should I stop sharing?
Yeah, if you want. Yeah, I think it's better. Then we can see other people as well. Okay, the first one is from Raja Nirujogi. And he says, Professor Bhalla, could you comment on or share your thoughts on the role of protein turnover in long-term memory maintenance? Mm-hmm. For example, long-lived proteins versus short-lived proteins.
Yeah, so that's a great question. In fact, that was the motivation for a lot of our work on this topic. So there's this great work by Mike Ehlers, which, among others, looked at these turnover times. And for most of the relevant synaptic proteins, the timeline varies from a few minutes to, at most, a few days. Now, you always have outliers, but the key molecules involved seem to be very, very short-lived. I mean, you don't want to trust your memories to something that lasts a few days. And that's the long-lived proteins. Now, there are other structural proteins in other parts of the system, but I'm thinking particularly about synaptic proteins, and those tend to turn over quite rapidly. Yeah, I mean, by and large, there may be a few exceptions, but it's not likely that they will be holding information for a very long time. So we think there has to be some kind of molecular switch to keep the information.
Right. Thanks a lot for that answer. Anjali Anil Kumar asks, are there any correlations between the size or structure of the hippocampus on the amount and type of short-term memories that are made or formed?
So this is a good question and has strong theoretical implications. So in brief, yes, there's very strong theoretical reasons for thinking that a large hippocampus or a large network with many synapses has a larger capacity. But remember that we're talking not about a neural network in the artificial intelligence sense. We're talking about a living organism. And one of the most important things that the organism has to do is to balance the need to remember with the need, paradoxically, to forget. Because you do not want to be hung up. You do not want to be thinking about what you ate for breakfast 3 days ago when you are confronted with an immediate decision. So it's very important for a live animal to not just form memories, but also to have some way to decide which is important and throw away the rest. So yes, there is a correlation in size, but that is far from the only important thing.
Right. Another question is from Shantari Gorai, who says, is there any change for Alzheimer's disease? I'm guessing she means, is there any change in memory formation mechanisms in Alzheimer's disease?
So, yeah, this is a question which, deeply affects many people who have aged relatives. And the answer is,
Yes.
there are major changes that happen. The most obvious and profound is that there is massive cell loss. But even before that happens, even before it's visible, there's both cell loss and synapse loss. And the thing that's really amazing, actually, is how resilient is the brain, how resilient is the circuit. That is, you actually don't even start to notice what's happening till some enormous fraction, like half or nearly that much of the connections are gone. So yes, there is a change during Alzheimer's disease, but remarkably, the brain does an extraordinarily good job of keeping things going despite that for a very long time.
Yes, indeed. Another question is, how are some memories strong and some not? Is it synaptic degeneration that's happening? So this is from DSS.
Yeah, so this actually again also touches on some of the key theoretical and behavioral aspects of it, which is that strength of memory is related to most to the context in which the memory is formed. So it's very well established that memories that are formed under very intense emotional situations tend to be very strong. So you will remember the major salient events of your life. You'll even remember many small incidents around those major salient events, but you won't remember everyday things like what you ate for breakfast even 2-3 days ago. Those sort of smear out. So yes, the number of synapses changes, but also the way in which memory stores changes. Things with a very strong emotional connection often have memory correlates in the amygdala in addition to the hippocampus. So there's many differences in how memories are formed when they are important and strong. And synaptic degeneration actually is not such a big part of that. This is more a case of how the memories are formed, which kinds of synapses they affect, how strongly they affect those synapses.
Right. I think there are quite a few questions, but in the interest of time, we are going to save them for later and have Dr. Poonam Thakur next. Thank you so much, Upi, for that fascinating talk, but we will come back to you at the end for the question and answer session, and maybe some of the audience will want to interact a bit.
Okay, thank you.
Thank you. So the next speaker for today is Dr. Poonam Thakur from the Indian Institute of Science Education and Research, Thiruvananthapuram. Poonam was at Punjab University, Chandigarh for her bachelor's and master's and remained there for her graduate work in the lab of Professor Bimla Nehru, working on the molecular mechanisms of Parkinson's disease, a research field that has fascinated her and that she has remained loyal to since. Following her graduate thesis, Poonam did her postdoctoral research first in the lab of Anders Borklund at the Wallenberg Neuroscience Center at Lund University, Sweden, and then at the Roper Lab at Goethe University from 2016 to 2019 in Frankfurt. Poonam returned to India on an early career Wellcome Trust India Alliance Fellowship and joined IISER-TVM as an assistant professor in 2020 in the midst of a raging pandemic, where she continues to solve mysteries about Parkinson's disease. And today she's going to tell us about her research in Parkinson's. So over to you, Poonam. The stage is yours. Welcome to Biologically Speaking.
Thank you, Shreya. Thanks a lot for the nice introduction. First question, am I audible and are my slides visible?
Yes to both.
Okay, perfect. So first of all, I would like to congratulate the team of Biologically Speaking for coming up with an excellent seminar series. I have been listening to some of the previous talks in the sessions, and they are always fabulous and informative, and I have enjoyed listening to them. So it's special to come on the same series, which I have been following for a bit. Okay. So to get started with my work, the title I gave is long and complicated and too specific. So before I take you all deep into all those specific things, let me first give you an overview of what I'm doing and why I'm doing all of this, okay? So as Shreya has mentioned, my forever love for research is Parkinson's disease. I have stuck to it since my PhD days and never ever left it. So what is Parkinson's disease? So it is one of the most common neurodegenerative disorders that's out there. With the increasing aging population, currently millions of people are suffering from Parkinson's disease. It is a well-known disease. It has been described in modern medicine for the last 200 years, but till date, we do not have any cure for it, unfortunately. The disease is characterized by a lot of symptoms, including non-motor symptoms such as problems with sleep, smell, and sometimes cognitive losses, etc. But the defining characteristic feature of this disease are the motor symptoms. So here you can see this picture of a man. You can see this man has trembling limbs, mostly hands and arms, they show tremor. Posture is stooped. The gait is problematic. Patients are able to take only small shuffling steps, which can lead to falls often. And also patients have problems with initiating movement or maintaining the movement often. And majority of these motor symptoms arise from the loss of neurons in the brain region called as substantia nigra. Here is a picture of a human nigra. And in black color, you can see these neurons of substantia nigra, which contain this neuromelanin pigment, which gives it its black color. But in patients with Parkinson's disease, you see here a very dramatic loss of these dopaminergic neurons. And this leads to lack of dopamine in basal ganglia, where the circuits are responsible for maintaining our good motor movements. Okay, and this is often accompanied by the appearance of protein aggregates, which are rich in protein alpha-synuclein. Alpha-synuclein is a monomeric protein in normal conditions, lies in an unfolded state or without any compact 3D structure. But under disease condition, it can form these amyloid aggregates, which are then resistant to degradation and accumulate into what we call as Lewy bodies. Alpha-synuclein is not the only component, but it's a very, very important pathological component of the Lewy bodies. Okay, so this Lewy pathology doesn't only show up in the dopaminergic neurons. In this picture, you can see here that this LP stands for Lewy pathology. This appears in a lot of brain regions, including brainstem and amygdala, olfactory bulb, even parts of hippocampus and cortex in later stages of disease. But the severity of Lewy pathology is not really proportional to the cell death that is observed. Here you can see that these dopaminergic neurons of substantia nigra, they are disproportionately affected by this pathology. On the other hand, other regions such as dorsal motor nucleus or vagus nerve, despite having high amounts of Lewy pathology, doesn't show much neurodegeneration. And this question has baffled scientists for a long time, that why is it that these dopaminergic neurons show such vulnerability to degeneration under the Lewy pathology? And lots of theories have been put forward in literature to try to answer this question. One of the most prominent being that dopaminergic neurons, they are very unique. They have very, large axonal arbors. In fact, one of the largest in the central nervous system, leaving the motor neurons, etc. So they have incredibly large number of synaptic release sites. And on top of it, the axon is also non-myelinated. Okay, so a single cell body has to take care of immense amount of synaptic connections, which places these cells under heavy metabolic burden. And that's not all. In fact, they also have very unique firing properties. So most of you know that neurons fire when they receive some synaptic input. So these neurons also show this synaptically triggered firing like this. But in addition to this, these neurons also show synaptically independent firing, which arises spontaneously. This is a low frequency, very rhythmic firing, and hence is called as pacemaker firing as well. And with each action potential, we know that a lot of calcium comes inside the neurons. Because these neurons are constantly firing these spontaneous action potentials, that means a lot of calcium is coming in with each spike. And this excessive calcium can lead to then a lot of problems in the neurons if not taken care of. But that means the cell is under constant stress to take care of this excess calcium, to take care of a large number of the synaptic release sites, and this huge axonal arbor to take care of. So this type of, all of these things are collectively called as stressful pacemaker theory, that the spontaneous firing puts these neurons under heavy metabolic burden. While a lot of this is true and to some extent explains, it still doesn't explain a lot of things about the high vulnerability of dopaminergic neurons. Okay, so dopaminergic neurons are not uniquely localized into the substantia nigra region. They are also found in many other areas, notably in this brain region we call as ventral tegmental area. It lies just adjacent to substantia nigra in the brain. And it also has all of these giant axonal arbor and all of this pacemaker firing. But still, it is a very, very well-known fact that VTA neurons are quite resistant to degenerate during Parkinson's disease. This is a coronal brain section from a mouse brain atlas. So you can see the brain areas clearly. So even within substantia nigra, median substantia nigra region marked here is more resistant to degeneration in comparison to this lateral substantia nigra region. So all of these share these stressful pacemaker phenotype, but still the vulnerability to degenerate is so different among these various subpopulations of dopaminergic neurons. And that's where my project started. And that's the question I wanted to answer, that why is there so much diversity among dopaminergic neurons in their response to pathology during Parkinson's disease? So to answer this question, I wanted to take the electrophysiological route, and I wanted to have a look at how these various subpopulations of dopaminergic neurons will respond if they are exposed to alpha-synuclein aggregates. So to do this, what I took was, there are no molecular markers that at the moment that can help us recognize these various cell populations, subpopulations of dopaminergic neurons. So to identify them, we had to rely on retrograde labeling. So these neurons project to large number of areas in striatum and in cortex, etc. And this is a very topologically organized region. So we took advantage of this thing. So to get to VTA, what I did was I did retrograde labeling with fluorescent beads in this region called as nucleus accumbens cores. So this region is receiving projections from VTA region. Then I also injected some animals in dorsomedial striatum. This is the region which is receiving projections from medial substantia nigra. And some injections were done in dorsolateral striatum, which is receiving these projections from lateral substantia nigra. And then I waited 3 to 4 days to let these beads travel back to the cell bodies from where the projections were coming from. And then I obtained the brain slices and I did patch clamp recordings from them. And while doing the patch clamp recordings, I applied alpha-synuclein in the solution that was contained in the patch pipette. This approach offered us the advantage that we could apply a very selected given amount of alpha-synuclein to each individual cell. And had we applied alpha-synuclein extracellularly, for example, in the solution in which the slices are kept, we would have run into the problem in like which cells would take up more alpha-synuclein and which cells would take up less alpha-synuclein, and that could complicate the differential response that we are interested in looking at. So we went with the application of alpha-synuclein through the patch pipette to have a greater control. And during the recording, I filled these cells with neurobiotin, so we could later confirm them with histology that the recorded neurons were indeed dopaminergic. So we stained them with the tyrosine hydroxylase, a marker for these neurons. And then we also confirmed that these contained these fluorescent retrobeads to confirm that they were indeed projecting to the right area, or we were picking up the right cell subpopulation of these neurons. So let's now look at how the firing pattern of these various subpopulations were. So first of all, I looked at these VTA neurons, which were projecting to the nucleus accumbens cores. This is a lower magnification picture of the VTA region, and in green here, or the shining green, you can see the neurons that I recorded and filled. And here are the current clamp traces of these neurons. For people who are not super used to look at the electrophysiology data, I'll just show you the blown up picture of the first 10 seconds of this trace. So this is what these individual action potential spikes look like. And on a compressed x-axis of 300 seconds here, this is how these action potentials are looking like. So these are the action potentials without any current injection. So these are the spontaneous pacemaker firing that we had. And you can see that when we expose these neurons to either the monomeric alpha-synuclein or the fibrillar form of alpha-synuclein, we saw no change in any of the firing activity. We quantitated them in terms of firing frequency, and we saw no change in firing activity in any of the treatment groups. We also looked at the average membrane potential. Here we had to use average membrane potential instead of resting membrane potential, because as you saw, these neurons are never at a resting state. We also looked at the regularity of the firing using coefficient of variance, which is nothing but the interspike interval divided by the standard deviation. So the smaller this number is, more regular the firing is, and the bigger it is, more irregular the firing is. So we can say that regularity of firing, which is the hallmark of this pacemaker firing, is also not affected upon application of either fibrillar or the monomeric form of alpha-synuclein. Next, we looked at the monomeric, at the medial substantia nigra neurons that are projecting to the dorsomedial striatum here. And here again, you can see some of the filled neurons which were recorded. In presence of monomeric alpha-synuclein, again, no response. But in presence of fibrils, some mild irregularity was observed. But upon quantification, we saw that this irregularity was observed only in few neurons, but majority of neurons that were recorded were unaffected. So overall, there was no significant change from the control populations. Monomers also didn't show any change. In addition, the firing frequency was not at all altered. Neither will we observe any change in the average membrane potential of these neurons. However, when we looked at the lateral substantia nigra neurons, we saw that monomeric alpha-synuclein, as before, didn't cause any change. But in the presence of fibrils, we could see a very, very dramatic response. Here you can see that very, very quickly that the firing starts to become irregular. And after some time, it completely stops firing. And this is also conjugated by a decrease in the membrane potential, which we call as hyperpolarization. So here is the quantification. You can see a very rapid and dramatic loss of firing frequency, a very clear hyperpolarization of the membrane, and a very obvious increase in the firing variability. So overall, it was clear to us that lateral substantia nigra neurons were showing a very dramatic loss or response to the alpha-synuclein fibrils. But since we were forcefully putting these fibrils inside the cells, to really confirm that the results we are observing are not a result of an artifact or anything, we went for a further confirmation of these effects. So for this, what we did was, we did another set of recordings of lateral substantia nigra neurons. But here we did on-cell recording. So we made a patch with the patch pipette. We made a seal with the patch pipette, but we didn't open the membrane. So we just recorded in the on-cell mode. And for the application of fibrils, we brought in a second pipette in immediate vicinity of these neurons and puffed them on to the cell while we were recording this neuron. So in that case, no force feeding, but just a response. And what we saw was that when a control solution was given, the cells showed no response to their firing and they continued to fire as usual. But when we exposed these neurons to fibrils, we could see a very rapid loss of firing activity as before. So overall, it was very clear to us that when we are exposing these neurons, various subpopulations of dopaminergic neurons to alpha-synuclein fibrils, the pattern of selective degeneration that we observed in Parkinson's disease patients in a process that lasts for decades can also be replicated in this very short-term experiment lasting only 5 minutes. So VTA neurons show absolutely no response to these fibrils, while medial substantia nigra neurons, some of the neurons showed some irregularity in firing in response to fibrils, while lateral substantia nigra neurons were dramatically affected by the alpha-synuclein fibrils. So it was very clear phenotype that we had, and the response was also very selective to the fibrillar form of alpha-synuclein, which is believed to be the pathological form, but not to the monomeric form of alpha-synuclein, which is a normal cytosolic, which is a normal protein that exists in cytosol and the synapses of these neurons. So the next obvious question for us was of course, what is the mechanism behind this? So the firing activity of dopaminergic neurons is regulated with the interplay of large number of ion channels. And among the several suspected channels that we had, one channel that caught our eye was K-ATP channels. So these are ATP sensitive potassium channels. And these consist of these pore-forming KIR subunits and the sulfonide urea receptor subunits, which are the regulatory subunits. So under normal conditions, these neurons are usually lying in a closed condition. So they open only under specific conditions when there is energy depletion or ATP depletion, or cell is under some redox, its redox status is changed, there is high oxidative stress or something. So under usual conditions, it is bound by ATP and maintained in its closed state. But when ATP levels are depleted and ADP levels rise, they go and bind to this regulatory subunit, leading to opening of this pore-forming subunit and leading to extrusion of potassium from inside of the cell to outside, making inside more negative, thereby reducing the firing activity and also reducing the ATP consumption. So this can help to reduce the neuronal activity and neurotransmitter release when the cell is under distress or has low ATP levels. So because we observed a very clear hyperpolarization in our lateral substantia nigra neurons, we wanted to see if the KATP channels were involved in mediating this effect. So to test this hypothesis, what we did was we repeated these recordings in the presence of glibenclamide, which is a selective inhibitor of KATP channels. So another set of recordings was done with fibrils, and we could again see the loss of firing activity. But when recordings were done in presence of glibenclamide, we can see that no loss of firing activity was observed even in the presence of fibrils. And this we could see in quantification that loss of firing activity was rescued. So was the membrane potential, and the firing variability was also dramatically reduced in presence of glibenclamide. So it was quite clear that K8P channel opening was likely playing a very big role in mediating the effects of alpha-synuclein fibrils onto these lateral substantia nigra neurons. But to further confirm this role, we performed another set of experiments by using these transgenic mouse, which had a mutant form of the spore-forming subunit KIR 6.2, only selectively in the dopaminergic neurons. And we use wild-type retermates as controls. So when we did the recordings in these control mouse, we could still see the loss of firing activity in lateral substantia nigra neurons in presence of fibrils. But when we did the same experiments in these KIR 6.2 mutant mouse, we could see that there is no loss of firing activity even in presence of fibrils. This is the quantification of the same thing here. And we could also see that there is no change in firing variability or in the membrane membrane potential when these channels are knocked out. So both of these results led us to believe that K8P channels were playing a central role here. And the next obvious question for us was, then what is the stress that has led to opening of these K8P channels, right? For this, we looked into literature, and there were large number of studies which said that what alpha-synuclein does is causes calcium level dysregulation. This calcium tries to then enters the mitochondria and causes high level of ROS production from there. And so we thought maybe this mitochondrial ROS generation is what might be leading to opening of these K8P channels. Okay, and if calcium dysregulation is what is causing this ROS production from mitochondria, what we thought of is like stopping this calcium from entering the mitochondria. So mitochondrial calcium unipotor is one of the major channels that is responsible for uptake of calcium from cytosol into the mitochondria. And what we did was we blocked this channel with the help of this compound called as Ru360. And when we exposed these lateral substantia nigra neurons to fibrils in presence of Ru360, we saw that there was no loss in firing activity. So this could also be seen in the quantification here. It rescued the membrane potential, firing frequency, as well as increase in firing variability. So overall, it was clear to us that most likely the mitochondrial stress is involved in activating the K8P channels. And if we block the calcium entry into the mitochondria, we can block the K8P, we can block the lateral substantia nigra neuron loss of firing activity in these neurons. Or if we block K8P channels, then also we can rescue these neurons. We are still on the lookout of how exactly the calcium level dysregulations can happen in response to alpha-synuclein. So what is more upstream and what led to this entry of calcium into the mitochondria in first place. But that is still under exploration. And with this, I would end this talk. And I would like to thank my postdoctorate lab of Jochen Roper at Goethe University, where majority of this work was done. And I would also acknowledge Kevin Luke from Pennsylvania, who provided us with fibrils and monomers for this study. Currently, I'm working at IISER-TVM. So I acknowledge the funding support from IISER-TVM and Welcome Plus DBT India Alliance. And last but not the least, my lab here. All the students in the lab, we are now working on various aspects of Parkinson's disease. We are a very young lab and we are just venturing into experimental work after recovering from the lockdowns and effects of COVID on us. And I am quite active on Twitter. So in case some of you would like to get in touch with me or would like to collaborate or discuss or brainstorm some ideas, I'm more than happy to get in touch with you. And I welcome any questions from you. This is a picture of my beautiful campus for you to look at while I answer your questions. Thank you.
Thank you so much, Poonam, for this fantastic talk. We have a few questions. Upi, if you can also join in the discussion, that would be nice. First question is from Shonak. He says, does alpha-synuclein fibrils directly bind to K-ATP channels?
Currently, we do not have an evidence for that. And that would need another set of experiments for which we or my postdoc lab do not have the expertise for. And we really need to find some help from other places to look into doing those experiments. But based on whatever experiments we had some expertise in and what we managed to do, looks like this is a response to other stresses which are upstream of this, because we were able to block that when we block the mitochondrial calcium. I wouldn't say that there is absolutely no binding, but this is also likely happening through other routes, as we could demonstrate.
Right. Another question is, great data, Poonam. Hassan Mohammed says this. In my understanding, the retrobreeds are very non-specific. So how did you account for spread, and how did you separate the neurons projecting to different regions? And just out of curiosity, does deep brain stimulation show similar effects in patients, or is it not studied at all?
I'm getting some background noise. Okay, yeah. To answer the first part of the question, so these retrobeats are, so there isn't a very 100% specificity in them. So there could be something, a bit of non-specific labeling as well. But what we try to rely on is as accurate injection as possible. So we are localized into a given brain area. And then the projections, then we go back and look at the region where we are trying to look. So for example, if I'm injecting in dorsomedial striatum, I'll go back and look anatomically in the medial substantia nigra region only. And if some projections, I can see some labeled cells which are outside of that region, I would try to avoid them during the recordings. And then I'm at least taking the specificity of that I'm indeed recording from dopaminergic neurons, and I'm indeed in the right region by combining as many things as possible to have as much accuracy as possible. But again, 100% isn't possible for us.
Right. No, I can imagine how difficult these recordings must be. Another question is from Ghanendra Singh.
Sorry to interrupt you. I think I missed out a second part of Hassan's question, which was about the deep brain stimulations. I think question is a bit generic in this sense. So deep brain stimulations are shown to show benefit in patients to a large extent, but they are done not in nigra, neither in striatum. They are done in thalamic regions usually. And the exact mechanisms by which they help is not very, very clear, but they seem to work. If you go and stimulate them, then those regions, patients can often show remarkable recovery in their motor features. But it's a highly, highly invasive procedure. You need to, drill a hole in your brain, and then you need to go also under your skin, somewhere usually in shoulder region, to put this battery to stimulate this pacemaker. Abroad, patients are still able to afford that, but in India, that surgery costs about 30 to 40 lakhs. And you can imagine the feasibility of getting that surgery for most of the patients who are old and from not well-to-do families is a very, very difficult possibility. So it's usually not a first route of action for majority of patients.
Okay, another question from Ghanendra Singh is, how are calcium levels kept at homeostasis under stress inside the cell or the synaptic cleft? Are there known molecular mechanisms in literature through which it occurs?
So usually, calcium can be taken care of by, there are a lot of these calcium buffering proteins. For example, calbindin is there, and many others. And my memory is really failing me at the moment. But there are few other calcium binding proteins which can take care of handling them. And also we have these intracellular calcium stores. For example, endoplasmic reticulum is a big store, and to some extent mitochondria. When calcium enters the cells, these calcium buffering proteins or these stores can take care of these levels. Problems start to happen when too much of calcium is entering, that these stores can be overwhelmed, or these calcium buffering proteins are not enough. Or in case of these more vulnerable neurons of substantia nigra, many of these neurons are actually lacking these calcium buffering proteins, or their levels are much lower. So for people who are looking at these differential vulnerability, there have been data in literature which shows that VTA neurons, for example, have high levels of calcium buffering proteins in comparison to more vulnerable lateral substantia nigra neurons, for example. But there are some populations which are mixed, which have this but are still vulnerable. So it always gets a bit complicated in case of this. So there is probably more factors to this diversity, which we do not understand currently. But yeah, there is a somewhat a huge range there. So in response to alpha-synuclein fibrils, a lot of people say that it gets dysregulated, but how exactly that happens is really a black box. I haven't really found so far any study which can tell us exactly how that dysregulation happens. And that is what I'm really interested in finding out, and going behind that, how exactly that process happens, we do not know.
Thank you so much for that detailed answer. If I can take advantage of my situation as moderator and ask you a question as well. So in Parkinson's, usually the aggregation of fibrils leads to a slow degenerative process over time, right? And
Right.
Here you're showing like acute application of fibrils sort of affects the activity of cells in vitro.
Right.
You have a speculation of how this happens? So this is one question. And second is, do you know, or is there literature of how the activity of these cells occur in vivo, or what they respond to in vivo?
Yeah. So that's really an excellent question. And we also wanted to ask that what happens, because this is in vitro, of course, it is devoid of circuitry. What happens if similar amounts are injected in vivo? Can we really see any outcome or of this? So we tried a lot with another set of experiments. So we did these. We took some mouse. We implanted this some cannula inside their brain, and which was into the nigral region, and then we tried to inject these fibrils through the cannula while they were performing behavior. And we tried to see that if there is a acute stopping of this activity, can this lead to any behavioral outcome in the short term that we saw, that we are observing in the in vitro slices. And unfortunately, we didn't see any of that. So at least in the very quick short-term experiments, we didn't see any change in the behavioral phenotype of the animals. So we tried with various doses and various variations, and it didn't happen like that. So what we are speculating is that in the in vivo systems, because brains are quite, they try to run a lot of compensations when something is going wrong. And that is one of the reasons that this neurodegeneration takes so much time to even be clinically diagnosed, right? Because you have this disease for several years, and by the time you start to show motor phenotype, you have lost more than 50% of your nigral neurons already, because brain was running a lot of compensation in that term. So I think in the in vivo, what we would expect based on this and this literature is that it wouldn't be as fast as we saw in vitro, because a lot of compensatory mechanisms might be kicking into the picture. And also cells might find a way when they are alive and all of their circuitry is there to diffuse these fibrils to some extent, or to metabolize or get rid of them to some extent. So cells would give a good fight before succumbing to it. In this in vitro scenario, cells didn't have chance to do any of these things. So it's a glimpse into the whole thing, but looking there would need more experimentation. Yeah.
Thank you for that answer. There are a few more questions. I think in the interest of time, we will take 2 more maybe. And then for the audience, if you have more questions, please feel free to write to the speakers or get in touch with them. And I would also ask Rupkatha to jump in with some questions which have been asked on YouTube as well. So I'll just take 2 more questions and pass on the field to Rupkatha. So there's a question for Upi actually. So Tamishra Pal says, Professor Bhalla, thanks for an excellent talk. I'm curious to know about how we can stop beta sheet aggregation process in Alzheimer's patients, and how effective are recent advancements in the prevention of the disease. And the second question, Upi, maybe you can take them both together, is regarding 2P imaging in the hippocampus and how difficult it is, and what happens if you remove the layers of the cortex when you go in with the lens.
Okay, so the first question about how to stop aggregation, I confess I don't have a clue. It's something that many people have looked at, and I'm certainly not among the people who's familiar with that area. Since so many people have tried and it still remains a problem, I imagine it's a very, very difficult challenge. The second problem about how difficult is 2P recording, well, it's neurosurgery. It is something that is very fine and delicate, especially because we're doing this at mouse scales. So you need, very much sub-millimeter scale precision in what you do. It's true that you have to, in order to image down to the hippocampus, you have to remove a little bit of the cortex. Fortunately, it does not seem to have a noticeable effect that we've found on the behavior of the animal. Again, because the brain is very resilient. But yes, it is a fairly severe manipulation, and it is conceivable that there are unexpected things happening as a result. But the animals are learning.
Thank you. With that, Rupkatha, would you like to jump in and ask a few of the questions that have come up on YouTube?
Yes, thank you. Thank you to both of our speakers for this exciting talks. It was a great Sunday evening we spent today. Ma'am, I would like to ask you a question from YouTube, which we have received. I think it refers a bit about your journey. How did you apply to foreign institutes, and your journey from Punjab University to becoming a mentor, and what would you say about this journey that would inspire younger students who want to take up science, wants to go into scientific research? And please tell us a little bit about your journey.
Okay. Yeah, well, that goes in a very different trajectory. But I can, only thing I can tell you is that it's all about your persistence and perseverance. I wouldn't say I'm extraordinarily talented or intelligent or anything, but I am extremely stubborn to things that if I want to do something, I will go and find a way to do something. That's pretty much it. I come from a family with agricultural background. I don't have any well-educated people in my family. Neither are my parents go to college or anything like that. So I really didn't have any guidance while I was in college. Not I had anyone telling me, these are the career options. This is how science can look like. You would be surprised to know that by end of my bachelor's, I didn't even know what NET exam is. Only when a senior cleared this exam and it was taught in the entire department, I came to know what a NET exam is. And there have been so many moments that when I look back now, I think, gosh, how stupid I have been. But only thing is, ignorance is a bliss. I didn't know at that time that I am being stupid. I just had a fool's confidence to go about, and I really went about and asked anyone who could tell me anything and who could help me with anything. Literally shamelessly, can you tell me this? Can you tell me this? I really went about like this in this whole world. And I have been really fortunate that I found so many good people on the way who were kind enough and patient enough to help this first 10% to really go about her journey and teach her a few things and help me succeed. And I would especially name my PhD supervisor, Bimla Nehru. We were at a small state university without a lot of facilities, but still the things she did for me and things she taught me is beyond comparison. And also my second postdoc mentor, Jochen Roper. He has been the fantastic and bestest of the mentors I have seen in my life. And if there is, so I have, because I have met so many good and generous people on my journey, I just try to imbibe them and try to do the same thing for my students and people I meet on the way.
That was amazing. Actually, I think that's going to inspire a lot of us, and that resonated so much with me because I didn't know about NET during my BSc as well. So it happened the same way for me. I heard it from a senior. So yeah, there are so many people who inspire us, and none of us can do it alone, right? And a similar question I think I would pose to Professor Bhalla. We would like to know a little bit about why you chose to do science and you chose to take it up as a career and something that would encourage younger students who are studying science at college and university levels, and what is it that they should keep in mind in order to pursue science, in order to keep the spirit of curiosity alive within them. Could you please share a bit about your journey?
Sure. Yeah. So I was hooked into science very young by my mother, who was a really inspirational person. She got me this old, I don't think they have it anymore, which is a shame, old series of thin little books about science called How and Why. And I devoured most of them. And so, yeah, she just kept giving me fascinating material like this. And I was very, very early sure I wanted to do science. And I just sort of followed that through. I actually had in mind to be a physicist. And at one point, I had to make the decision between doing fusion physics, plasma physics, versus doing biology for my PhD. And biology won by a small margin, and hence things have followed from there. Just like Poonam says, it's a lot of sort of random blundering around with a lot of good fortune and extraordinary people who have been mentors at different points in my career, and a lot of great good fortune. I mean, to myself, I would just say, you have to, for my own perspective, you just have to love what you're doing. The science has to be a motivation to you, even when there are downs, which there are, and then you can enjoy the ups, which come every so often. But it's you just have to persevere and enjoy doing the science. The everyday parts of it, all the small things are the rewards. If you're just hung up on getting the great paper or accolade or something, then you'll not get anywhere. You just have to enjoy the process of doing science.
Enjoying the process. That's very well said, and I hope that's going to be as inspiring for all the viewers and listeners we have today as it was for us. It was amazing to have you both today, and thank you so much for answering these questions. Shreya, would you take over, please?
Yes, thanks, Rupkatha. Again, thank you to the speakers. Thank you for accepting our invitation and for those really illuminating talks and taking time on a Sunday to be with us and chat with us for so long. With that, we would bring the session to a close. Please subscribe to our YouTube channel, but also please follow us on Twitter because we have a lot of exciting events coming up. We're celebrating CRISPR Day on October 17th or 18th. We will have announcements coming up about the webinars, but also in a month and a half, in November, the 6th and 7th, we are celebrating International Science Day with a virtual science fair that is directed towards science young enthusiasts from the level of, let's say, high school to undergrads and early graduate students. So please stay tuned for all of these announcements and exciting events that are coming up on Biologically Speaking. I thank all the team who are behind this, helping us out as well. So have a nice Sunday, and this is goodbye from us here.